Hello, today I'm going to talk to you about arteriovenous malformations in the brain. Okay, so in this photomicrograph, you can see fragments of tissue that consists of multiple variable size and shapes of thin wall blood vessels. So if as you go on higher magnification, you can see there's a variability in size and shapes of dilated blood vessels that are filled with red blood cells. So here is the highest magnifications of the photomicrograph of the fragments of tissue. You can see um, there's a lot of size. There's a variability in size and shapes, and in between these vessels, there is a very minimal fibrous fibrocollagenous stroma. So here is another fragments, but these fragments of tissues is a little bit different compared to the previous fragments. Here you can still see there's a variable size and shapes of dilated blood vessels, but in between these blood vessels there are some tissue. It's not just a fibrous tissue, but it's a different type of a tissue. So on higher magnification, you can see that these vessels, all of these are vessels that are filled with red blood cells. There is a brain parenchyma or a type of tissue in between these vessels. So I make a labeling here just to demonstrate that in between these vessels actually brain parenchyma that are showing features of gliosis. They increase in cellularity, and the astrocytes of the glial cells will become a little bit plump. But this is not a fibrous tissue or not a fibrocollagenous tissue in between them, but it is a more of a brain parenchyma. So I did some EVG staining or the elastine stain just to demonstrate the variability of the types of the vessels. At the first tissue fragments, you can see the vessel is made up of a very thin wall kind of a vessels. It doesn't have any smooth muscles or a thicker fibrocollagenous wall. It's just like sort of capillary types of vessels. But in a few places, especially in area that contains brain parenchyma in between the vessels, you can see there's a thick wall vessels. In these instances, it more of like arterioles or venules, small arteries or small veins kind of vessels, where the the vascular wall is a little bit thicker compared to the initial vessels or in the first fragments. So this is just to demonstrate the thickness of the vessel's wall. So here is another example. This is another thick wall vessels where the vessel the width of the thickness of the vessel wall is a bit thicker compared to the first or the capillary like kind of vessel. Here is another tissue fragments. I'm taken from another area. You can see again a lot of vessels, dilated blood vessels. They are separated from each other by brain parenchyma in between them. So all of this is the blood vessels that are filled with red blood cells. So in between them, you can see there's a brain parenchyma. And here is another interesting findings where this is actually a ruptured arteries. This artery is a bit bigger compared to the previous arterioles, and you can see that this arteries wall is at some area is ruptured. So it's ruptured here, and it spill out all of the blood and also the fibrin. You can see all of these are actually fibrin and red blood cell or laymanly we call it as blood clots. So you can see the blood clots already being extravasated from the vessels due to the arteries rupture. So here is another area just to demonstrate that the arteries, the vessels wall is composed of intima part and also the smooth muscle part where the tunica media is. So this actually is the arteries wall. So I also did elastin stain on these fragments, and just to demonstrate the artery wall here, you can see there's a thickness of the tunica intima of the blood vessels or the arteries. So in view of the first fragment that shows a little bit features, sort of a cavernoma like features. So I present to you some differences between AVM and also cavernoma, because in the first fragments the features is more or less like a cavernoma, but there's some differences there. For AVM, you can see there's a variable type of vessels. Like in this case, we can see arteries and also veins and also capillaries. But in cavernoma, it's more of a honeycomb-like. The vessels is arranged in a honeycomb-like features or structures, and it's composed of a very compact vessels or a very compact thin wall vessels. And in AVM, you can see there's a surrounding stroma is a bit less sclerotic, like in this case. But in the cavernoma, the surrounding stroma is very densely collagenized and also sclerotic. And the most important thing, you can see the brain parenchyma in between the vessels in AVM, whereas in the cavernoma, because of the compact arrangement of the vessels, you cannot see any brain parenchyma. So they're just a very dense collagenized and sclerotic stroma in between the vessels. So by that, thank you. You can follow me on Twitter and also Facebook and also Instagram. By that, thank you.